guys, let's do section 4, the cell cycle and cancer. It's going to be super exciting, I can feel it. Abnormal growth of a cell is called a tumor. Benign tumors are not cancerous. They are encapsulated, meaning they have a layer around them, and they do not invade neighboring tissue or spread. Malignant tumors are cancerous. They are not encapsulated. They don't have a layer around them. They readily invade neighboring tissues. They may also detach and lodge in distant places, which is called metastasis. I have trouble saying that word, guys, sorry. And this results from the mutation of genes regulating the cell cycle. So the development of cancer tends to be a gradual process involving a lot of steps. And it may take years before the cell is obviously cancerous. This might be TMI, but I had a mole on my wrist removed about a year ago, almost a year. If you've ever looked at my right wrist, you'll notice a scar there. That's where it was. It was not cancerous, but it was precancerous. So it was on its way to developing. It was in this multi-step pathway to the development of cancer. So my dermatologist wanted to get rid of it before it caused any further problems. Characteristics of cancer cells. They lack differentiation, okay, meaning they don't have a specific job. They're not specialized. And they can be immortal, okay, not meaning that one single cell will live forever, but it can enter the cell cycle repeatedly. Okay, most cells, they only do it a certain number of times, and then they're like, oh, this is my last one. Good knowing you. Another characteristic is they have abnormal nuclei. The cells might become enlarged, and they may have an abnormal number of chromosomes because the copying process doesn't work correctly, or the division might not work properly. And they often have extra copies of certain genes. They do not undergo apoptosis, which you'll remember is programmed cell death. Normally cells with damaged DNA undergo apoptosis, but in cancerous cells, they don't recognize that. They don't recognize that the DNA is damaged, and they just keep going and going and going. Okay, cancer cells are abnormal, but they fail to undergo apoptosis. Okay, so they're damaged, they aren't working correctly, but apoptosis is not working the way that it should to put a stop to that. Characteristics of cancer cells. They form tumors. Mitosis is normally controlled by contact with neighboring cells, something that is called contact inhibition. Cancer cells don't have that. They also lack differentiation. They are non-functional in their tissues, so they don't do their job right. They have enlarged and abnormal nuclei, and they undergo metastasis. The original tumor easily fragments, it easily breaks apart and forms new tumors in other organs. And they undergo angiogenesis, where they form new blood vessels. So they attach to an organ, and they develop new blood vessels to bring more nutrients and more oxygen to the tumor so that it can grow faster. And it doesn't undergo apoptosis. So this is just a horrible combination, causing cells to go crazy. They don't have apoptosis correctly, they break apart, and they can reroute blood vessels so that they get more nutrients. Here we have one bad cell right here, this little dark brown one. Okay, that has a mutation, that has something wrong with it. It has the ability to start a tumor. So it replicates and makes more and more and more. Okay, this would be like the mole on my wrist that I had removed. And, oh, we've got one that is truly cancerous. Okay, it probably took a long time for these brown cells to develop, okay, to have that many of them there. And over time, one of them becomes cancerous, and it doesn't have the gene, it doesn't have the signal that causes apoptosis. So it keeps dividing and dividing and dividing and dividing until it attaches to a blood vessel or a lymphatic vessel. Those Your lymph system carries lymph, obviously. It's like plasma in your blood. Okay, and once the cancer invades that blood vessel or that lymphatic vessel, it can easily move throughout the body and cause tumors elsewhere. This is how we get metastatic tumors from the primary one. Okay, they invade the lymph system and they move throughout the body. Here we have a chart describing the difference between cancerous cells and normal cells. Normal cells are differentiated. They have normal nuclei. They undergo apoptosis. They have contact inhibition, and they're usually very organized in layers, and they remain in the original tissue. Cancer cells are the exact opposite. They are non-differentiated. They have abnormal nuclei. They don't undergo apoptosis. There's no contact inhibition. They're very disorganized and multi-layered, and they can metastasize. The origin of cancer. Normal growth and tissue maintenance depends on a balance between the signals that promote and inhibit cell division. There are two types of genes that may be mutated when the balance is upset, which may cause cancer. Onconogenes, proto-onconogenes, I'm not sure if I'm saying that right, sorry guys. They code for proteins which promote the cell cycle in various ways. If it becomes mutated, it becomes an onconogene. Tumor suppressor genes code for proteins which inhibit the cell cycle and promote apoptosis in various ways. If a tumor suppressor gene becomes inactive, it may promote cancer development. Both of these genes are normally regulated in coordination with an organism's growth plan. These directly inhibit the cell cycle and prevent the cell from dividing uncontrollably. Some tumor suppressors promote apoptosis, and mutation in a tumor suppressor causes the cell cycle to accelerate. So it's stopping that apoptosis. Examples are the RB and P53 genes, which code for proteins with the same names. Retinoblastoma is an inherited condition that results from a mutation in the RB gene, and P53 gene turns on the expression of other cell cycle inhibitory genes. Half of human cancers involve an abnormal or deleted P53 gene. Proto-onconogenes are normal genes which are part of a stimulatory pathway. They promote progression through the cell cycle. They include receptors and signaling molecules. Mutations in these genes cause them to become onconogenes. These can specify an abnormal protein product or produce abnormally high levels of a normal product. Uncontrolled cell division results from this. There are 100 onconogenes which can lead to tumors. Examples are the BRCA1 or BRCA1 mutations, which can cause breast and ovarian cancer. A few years back, there were lots of people that were getting genetic testing to see if they had this BRCA1 gene. And many of them made the choice to have their ovaries removed or have their breasts removed because the prevalence of cancer in people with the BRCA1 gene is incredibly high. Okay, someone that I used to work with did this. She found out that she had the BRCA1 gene. Many women in her family had died of breast cancer, and she made the choice to have her breast removed and reconstructed because the risk of cancer in her family was so incredibly high. Here we have a diagram showing the causes of cancer. Okay, it can be caused hereditarily. Okay, some of us just have bad genes. Some of us are exposed to radiation, and that can cause cancer, especially skin cancer. That's why it is important to wear sunscreen. I am particularly aware of this. I've had many people in my family develop skin cancer. So anytime I'm going to be outside for a long time in direct sunlight, I wear sunscreen, I wear protective clothing, because skin cancer is very prevalent in my family. Some people are exposed to chemicals that can cause cancer. cancer, like herbicides in the past, and viruses. There are some viruses that can cause cancer. Other causes of cancer, telomeres. Chromosomes normally have a special material at each end called a telomere. They are a long strand of thymine at the end of a cell. Not the end of a cell, sorry, the end of a chromosome. And with each cell division, they get fewer and fewer. And when they get too short, the cell just stops dividing. Telomerase is an enzyme that maintains the length of those telomeres. Mutations in that gene cause the telomeres to continue to grow in length, which allows cancer cells to continually divide because the DNA, the telomeres, are not getting any shorter. So the cell thinks, well, we can divide all we want, and they do. And that's it, guys. If you have any questions, let me know.